All right, let's dive in. When you picture DNA, you probably see that classic double helix, right? Maybe floating nicely in a test tube.
Mm-hmm. Nice, clean solution. That's definitely the standard lab image.
Exactly. That's how we usually study it. But what happens when you take that DNA and put it somewhere that looks, well, a lot more like the inside of your cells?
Which is, spoiler alert, incredibly packed, not clean at all.
Right. And that's the core question tackled by this really interesting study from Northwestern University we're looking at today. It sort of shakes things up.
It really does. So our mission here is to unpack this research because it shows just how much that super crowded, messy environment inside a real cell affects, the basic mechanics of DNA.
Things we thought we maybe understood completely from those simpler lab tests.
Precisely. It suggests the lab conditions might be missing some pretty crucial details.
And the headline finding, what's the big takeaway?
Well, the big one is that separating the two strands of DNA, which is fundamental for, everything life does, copying DNA, reading genes.
Yeah, super important.
It might actually take a lot more physical force when it happens inside that crowded cell compared to, the easygoing lab setup.
Wow. Okay. More force needed. And this comes from work by Professor John Marco and Parth Desai.
That's right. They're the researchers behind this recent paper. It really highlights this difference, studying molecules alone versus in their natural crowded home.
So let's contrast those two worlds a bit more. The lab versus the cell. Paint that picture.
Okay, so in the lab, like you said, it's usually DNA sitting in a dilute, mostly water solution. Pretty simple.
And often using heat to separate strands.
Yeah, often high heat, like over 150 Fahrenheit sometimes, which obviously your body doesn't do. It's very controlled, almost empty, around the DNA molecule itself.
Which is just fundamentally not what it's like inside a cell.
Not at all. Inside a cell, DNA is swimming in this thick soup of proteins, RNA, sugars, salts. It's unbelievably crowded.
So no high heat separation there. How does it happen?
It's done mechanically. Specialized proteins, helicases are a key example. They actually bind onto the DNA and physically pry the strands apart. It's an active mechanical process.
I liked Professor Marco's analogy for this. He described the inside of the cell as super crowded, like you said, and pictures all those extra molecules as being like billiard balls constantly pounding against the DNA.
Yeah, it's a great visual. And that pounding, that constant bumping is central to what they call the crowding effect.
Okay, so what does that actually do? How does it make strand separation harder?
Well, there are a couple things going on. Part of it is what's called an entropic effect. Basically, all those other molecules take up space.
Right.
So there's just less room, less available volume for the DNA strands to physically separate and move into. It sort of statistically biases the DNA towards staying closed, makes it harder for it to find that open state.
So the crowd just gets in the way, physically blocking it.
Yeah, physical interference is a big part of it. But there's another, maybe more subtle effect, too. These crowding molecules can actually pull water away from the DNA.
Dehydrate it slightly.
Exactly. They compete for the water molecules. And water is really important for DNA's structure and flexibility. So if you pull some water away, you can change the DNA's mechanics, maybe make it a bit stiffer, less willing to unwind.
So it's both crowding and potentially dehydration, making it tougher.
Right. The two effects can combine to stabilize the double helix, making it resist being pulled apart more strongly.
Okay, measuring something like force at that tiny scale inside a mimicked, crowded environment. That sounds really challenging. How did they actually do it?
They used a really cool technique called magnetic tweezers. Professor Marco was actually one of the pioneers of this method like 25 years ago.
Magnetic tweezers. What do they do?
So they attach a tiny magnetic bead to one end of a single DNA molecule. Then they use magnets to apply a very, very precise pulling force on that bead.
Gently pulling the DNA strands apart.
Exactly. And they can measure exactly how much force it takes to unzip the DNA strand by strand. It's incredibly sensitive.
And they didn't just make a generic crowd, right? They used specific molecules.
Correct. That was clever. They added different things like glycerol or polyethylene glycol, common lab chemicals used to mimic crowding.
To see if different types of crowding mattered.
Yeah, because some of those molecules might mostly cause physical crowding, while others might be better at pulling water away, causing that dehydration effect. So they could start to tease apart the different contributions.
And the key thing is they could quantify it, put a number on the extra stress.
Precisely. It wasn't just it's harder. They measured how much harder, how much extra force was needed because of that crowding.
Okay, so stepping back, why does this fundamental discovery matter? Why should, our listeners care about the force needed to unzip DNA.
Well, this kind of really basic research, understanding the physics of biology, it's often the foundation for huge breakthroughs down the line.
Like what?
Professor Marker mentioned how early fundamental studies on DNA mechanics were crucial for developing things we now take for granted, like rapid genome sequencing, the ability to read a whole human genome in under a day. That builds on this kind of basic science.
Right, okay. And thinking more directly about what happens in the cell, if crowding makes strand separation harder, what else might it affect?
That's the really big implication, I think. It's probably not just about strand separation. Think about everything else that interacts with DNA.
Like proteins binding to it.
Exactly. Proteins that need to read genes, repair damage, switch genes on or off. They all need to physically access and interact with the DNA helix.
And if the DNA is stiffer or harder to access because of crowding.
Then that changes the whole game for those proteins. It might cost more energy for them to bind, or maybe they bind differently or slower. It means our models for how all these crucial cellular processes work, models often based on dilute solutions, might need a serious update to reflect the crowded reality.
Wow. Yeah, that really broadens the scope. It suggests we might have been overlooking a key environmental factor.
It really does seem that way. And, they're pushing this further. Their next steps are looking at more complex mixtures of crowding agents.
Getting even closer to the real cell environment.
Right. And then starting to look directly at how specific enzymes, proteins that work on DNA, how their actual activity is affected by this crowding.
So, wrapping this up then, the core insight is pretty clear. Studying biology in a simplified test tube, it misses things. Important things.
Absolutely. You can't fully grasp how these molecules function without considering their actual messy, crowded neighborhood.
And the main takeaway for everyone listening.
That crowding inside a cell isn't just passive background fluff. It's an active physical force. It changes the energy landscape. It changes the mechanics, especially for DNA.
Which leaves us with a pretty fascinating final thought, doesn't it? If crowding affects DNA this much, how many other biological processes, proteins folding, molecules finding each other, reactions happening, processes we've mostly studied in simple lab conditions, how different might they be inside the incredibly packed, bustling world of a real living cell? It makes you rethink the whole environment.